will continue with our experiments and investigating photosynthesis. Today I would like us to concentrate on light and carbon dioxide. And now we are going to start with a few new investigations. The 4 investigations that will be important to us will be: is light needed for photosynthesis? Is carbon dioxide needed for photosynthesis? Is it necessary that chlorophyll must be present? Is it needed? And then, is it oxygen that is produced during photosynthesis? Today, we are only going to concentrate on 2 experiments, and that is light important for photosynthesis, and is carbon dioxide needed for photosynthesis? Just to help you to remember, where is light important? We remember that photosynthesis takes place into 2 phases. Yes, we have phase number 1. Phase number 1 is the light-dependent phase, a phase that cannot take place if light is not present. And in today's investigation, we are going to see, is it really the matter? We call it photolysis because this light will split the water molecules into the hydrogens and the oxygens. Now, and then we are also going to look at the second phase where carbon dioxide is reduced to form carbohydrates. That is the light independent phase. This phase does not need light. But as we said yesterday, if the light phase did not take place and there are no hydrogens coming into this phase to have hydrogens, carbons and oxygens that will form the carbohydrates, this phase will also not take place. Now, and that is what we are going to study today. So, we said, for investigations to take place, we will need 2 groups. We can only take 2 plants, but that is a bit risky. The more examples that I have in an investigation, and I can work with an average, the more accurate will my results be. But I will have 2 plants. The one will be called the experiment, where I give everything that is needed, in this case for photosynthesis. So I will need water, I will need the correct temperature, I will need chlorophyll, I will need carbon dioxide, I will need light. If I am investigating light, then I will remove the light in this experiment. In the control, I will give everything, even the light. So, why? I want to compare the 2 investigations, and I want to prove that if the light is removed, no photosynthesis takes place. You agree? Good. So, we also said, I have to destarch before I start the experiment. Can you quickly tell yourself why? What is the answer? Is this your answer? A plant is left in the dark for 24 to 48 hours. That is destarching. That all the stored starch is used up. I want to prove that the starch that was formed during the experiment was due to the factor under investigation, or was lacking, starch was lacking due to the factor under investigation. And of course, before I start with the experiment, I do the starch test to prove there's no starch in this plant. Now I have my control and my experiment, and I now have something to prove. Okay, and now let's look at a setup experiment investigation about, is light really necessary for photosynthesis?
Now in the next practical that we can do, the aim of the practical is to see whether light is indeed necessary for photosynthesis. Now, what would we need? We would need a destarched potted plant, a plant that has been left in a dark cupboard for about 48 hours, and you will see that we have now placed foil over parts of the leaf, blocking out light under the foil. You can decide exactly what pattern you would like to make. What you would also need is all the apparatus that you have used previously to test for the presence of starch in a green leaf. The method of this practical would be, once you have destarched the plant and covered parts of those leaves, you would allow this pot plant in the sunlight for at least 4 to 8 hours. Once we have done that, I would remove a leaf from that pot plant and repeat the test for the presence of starch in the leaf. When doing this practical, it is important to emphasize what the result of this practical will be. After testing for the presence of starch, we will see the following. After doing the test for starch, the last step in the test of starch is when we place iodine solution onto the pale leaf. On this side, we will see that there was an immediate positive color change. Iodine solution changed to blue-black. But on this side, you can see that iodine solution stayed the brownish color. Now, when doing this practical, our aim was to see whether light is necessary. So this part of the leaf that was left uncovered would be the experiment, but the part of the leaf that was covered by foil would then serve as the control of this experiment. Once we have our results and we've observed that when light was allowed, we saw that a positive color change occurred, and when light was not allowed, the covered parts did not change color. We can now take those results and see that our conclusion would be light is definitely necessary for photosynthesis.
So, grade 10, we have a covered part and we have an uncovered part. And you have to now make a label diagram to show the results. And it is so important that you make sure when you do this drawing that your drawing looks exactly the same as the drawing that they gave you. Okay? So I will then make sure that that part that is covered is also the part that I have covered there. Now, and now I will have to add labels. Okay? And of course we do this with a ruler. And now this part was covered. So this part will be yellow-brown. And this part was blue-black. And this part was blue-black. And now, as the Tannie told us in the video, which part is the experiment? The part where the light was taken away. So, if we use a highlighter and we say the experiment will be this part. Yes? That part is the experiment. Because we said this part had all the requirements for photosynthesis except the factor that is under investigation. Good? And then we have the control, and this will be the control. Why do I say so? Because the control undergoes the same as the experiment, except that the independent variable is kept constant. Oh, so why are we talking about now about the independent variable? I just want to explain it to you like this. If light is there or not there, and I draw a graph, I will have light on the x-axis because the light can be changing from very low to very high because light is what I want to show as the factor. So in our previous slide we say except the factor that is under investigation or the independent variable. Light was in this case our independent variable because it was there or it was by my control taken away. And that is why we say it is the independent variable. I hope that you do understand that. Right. You have experiment number 2 on page 161. I want you now read through that experiment and see there are a few questions. Now, question 6 and 7. Answer that in your book. Make a heading, experiment 2, page 161, the day after day. And it is about photosynthesis experiments. And answer that. Now, we are going to continue to look at carbon dioxide is necessary for photosynthesis. Okay.
During this practical, we are going to see whether carbon dioxide is necessary for photosynthesis. Now, what we need is we need 2 destarch pot plants, and to do that we need to place these pot plants into a dark cupboard for 48 hours. Then test one of the leaves out of each of these pot plants to make sure that all the starch has indeed been used up and that there is no starch present in the pot plants at that stage. And then you place these pot plants into a bell jar. Use pot plants that are more or less the same size in the control, we have soda lime in the funnel, and in the bell jar, sodium hydroxide in the beaker, and both these absorb all the carbon dioxide and make sure that no carbon dioxide actually enters the bell jar. On the other side, we have the experiment. In the funnel, we have normal chalk, and then inside the bell jar, it has water. The little beaker has water, but it could also contain calcium carbonate so that we do have carbon dioxide available so that this plant can indeed photosynthesize. Now we place these two bell jars into the sunlight for about 48 hours, and then you are ready to test for the presence of starch in both of these pot plants. The result of one of the leaves taken from the control, we will see that the leaf stays a yellow-brown color, which indicates no starch is present in the control. But in the experiment, we see that the leaf has turned to a blue-black color, indicating the presence of starch. Our conclusion will thus be that carbon dioxide is indeed necessary for photosynthesis, because we saw that starch is present when carbon dioxide is present.
Okay, so let's look at the experiment. Soda lime prevents entry of carbon dioxide. I have a bell jar, and normally we can even seal them by having Vaseline, petroleum jelly or Vaseline added here. Added here at the bottom, so to make sure that it is really sealed off. And we will do everything on both investigations. What do we see here? We see that they seal off even the soil. Why? Because remember, in the soil, there are a lot of microorganisms. And these microorganisms respire, and they will give off carbon dioxide. We want to make sure that there is no carbon dioxide in this jar. Good. I think that this is then the experiment, because our book says the experiment is where I have all the necessary factors for photosynthesis except the one being under investigation. Okay, so now I want you to concentrate on, why did I put in sodium hydroxide? Because all hydroxides absorbs carbon dioxide. So even if there was any carbon dioxide in this air, it would have been absorbed. Good. Soda lime absorbs it there as it moves. If there are any left in the air, absorb there, prevented from the microorganisms to release any carbon dioxide. On this side, we have the air coming in, and we know air contains carbon dioxide. Now, here in this picture, there's water, but I can add a carbonate, any type of carbonate in here, which will release some carbon dioxide to make sure that this one is filled with carbon dioxide. According to our book, the control is then this one. Why do I think this is the control? Because all the necessary factors, even the one that is being under investigation, is present here. Do you understand this? So here I added some more viewpoints and some more small important stuff to make sure this one has no carbon dioxide. All hydroxides absorb. Hydroxide absorb carbon dioxide. And in this one, there's water, but I can also add a carbonate solution there that will release some carbon dioxides. This one is the control because it contains all the necessary information. This one is the experiment because it has all the information except the one under investigation. Good. And that brings us to the end of this lesson. Answer the questions on experiment 2 and 3 on page 161. And please look, I also add a PDF with some good questions. On it, I want you to also try those questions to help you to practice and investigate photosynthesis.
There's no price like the ShopRite price, which means you'll get low prices on everything you need. Like 4 kilos Real Good frozen mixed chicken portions for only $154.99 and a 2 kilo bag of nice parboiled rice for just $26.99. The ShopRite price, the price you can trust, always.
This morning, the investigation on photosynthesis. And like we did in the previous lesson, we are going to first look at why. Today we are going to look at chlorophyll and is oxygen released during photosynthesis. So, again we are busy with, there are two phases. The first one is the light-dependent phase, during which the reaction is photolysis. And the second one is the light-independent phase, where the reduction takes place. Now, in our first experiment, we are going to see whether chlorophyll is important so that photosynthesis can take place. And in the second one, we are going to look at, is it really oxygen that is produced during photosynthesis? So both the investigations today will be part of the stage 1 light-dependent phase of photosynthesis. Good. The first experiment is then where we are going to look at, is chlorophyll necessary for photosynthesis?
In this practical, we will determine whether chlorophyll is indeed necessary for photosynthesis. For the practical, you would need a destarched leaf which is variegated, which means that there are parts that are green, which contain chlorophyll, and other parts, if you look on the margin here, you will see that no green pigment occurs in that leaf, and that's why it is colorless. Now, what do we need to do to destarch this pot plant? We would have to place it into a dark cupboard for about 48 hours to ensure that all the starch that has been produced previously will have been used up, even the parts that would maybe be in the non-colored parts of the leaf. Once we have left this pot plant outside for a few hours, I will then pick one of the leaves and I will test for the presence of starch as shown previously in the first practical demonstration. Now, once we have done that, and we have tested for the presence of starch, and we have added iodine solution to the leaf after the test for starch, we will see that the parts that did contain chlorophyll has now tested positive for starch. But on the margin, where we see that no chlorophyll was present, we will see that the iodine solution has stayed its brownish color. The middle of the leaf, if we compare it to this leaf, shows that the parts that contain chlorophyll have turned to a darker color. Now, I have placed the iodine solution onto this leaf, and the longer I leave it, it will actually turn even darker. So we can see a definite difference. The longer the iodine solution stays on that leaf, it will turn even darker to a blue-black color.
So we have seen a variegated leaf. Here on our premises are also variegated leaves. Exactly the same plant that I have here on the slide. So, if I have all those different leaves, we saw that the green parts are not always exactly the same. So it is very important to start your investigation by making a little drawing of your leaf and then just make an indication of exactly where was the green part and where was the white part. So that afterwards, when you do the test, you are able to prove that afterwards, now I have the blue-black parts on that specific area and the yellow-brown was on this area. And now I can compare this two and say, okay, the green tested blue-black, and the green was where the chlorophyll was. Now, which one would you say is the experiment? The experiment will then be the white part, because in the white part there was no chlorophyll, and the control will then be the part where all the necessary factors were available. Right. Our last one is oxygen really produced during photosynthesis. Okay?
Release of oxygen in photosynthesis. An Elodea plant is placed in a beaker filled with water and is covered with a glass funnel. A test tube filled with water is placed on the setting in a way to cover the tube part of the glass funnel. With the influence of light, the Elodea plant photosynthesize and produce oxygen bubbles. The oxygen bubbles move from the funnel into the test tube and collect here. In order to observe that the collected gas is oxygen, a burning match is brought near the test tube. The expansion of the flame shows that the collected gas is oxygen.
Good, grade 10. We normally say the test for oxygen is a glowing splint will reflame. Nah. So they just say the flame is going to burn up, but it's more or less the same. I hope that you realize we are now on page 162, and there you will find experiment 4 and 5. Please read through them and do the questions in your book. Then I also want you to look at the PDF that I added to this lesson with some more interesting practicing for you to get all the information that you need about investigating photosynthesis. Take care, grade 10. Look after yourself and stay warm, stay healthy. Something else that is interesting about plants, the type of nutrition that they use makes them a bit more special than the way we are just eating. Good morning, grade 10s. I hope that you had a nice breakfast. There was perhaps a few plant products. Yes. Chocolates? Yes, it's coming from plants. That you had bacon and eggs? Yes, it is also coming from plants because the little piggy needed to eat some plant food to make the nice bacon. And the egg will not be there if there was not a few maize kernels for the chickie to eat. So yes. What am I trying to tell you? I'm trying to tell you that plants are autotrophic feeders. Auto, self. They make their food self, but not only for themselves, for all the heterotrophs. They make the food. In order to do that, they can not only rely on the sunlight, the carbon dioxide and the water for the process of sunlight, carbon dioxide and water in a chloroplast is for, yes, photosynthesis. But they need to get minerals in order to build us all the food, nutrients, molecules that we need. And sometimes we can see on a plant that the plant is deficient of a specific mineral. Like if we have weak teeth, it can be calcium. If we have a large lump here, it can be goiter due to iodine deficiency. You remember that? Or if I have a lot of sores and weak bleeding gums, vitamin C. That is deficiencies. If I look at a plant and I see that there are discolorization on the leaves, oh yes, what discolor our teeth? Too much fluoride. So too much can also be a problem. But discolorization of the leaves may show a phosphate deficiency. And there we see only the veins are green. There we see the top part of the leaves are green and the rest are still looks green. And so we can see many different deficiencies in a plant. Now, it is important to go and study what is what. That's what we need to do. But let us link it to what we know. If we think nitrogen. Now we know that nitrogen is the N. Yes? Let me just get a pen for us. Nitrogen is the N. And the N is in which nutrient very important? In the proteins. So the nitrogen is for proteins. And interesting, if I have a plant, there's the roots, the leaves, and we also have a nice flower on this plant. It is important to note that the minerals can only come from the soil, especially nitrogen. We know that nitrogen is 78% of the air, but the plant cannot take the nitrogen from the air. No, the plant can only take the nitrogen from the nitrogen gas needs to move to the soil. And in the soil, it becomes nitrates. It dissolves in the soil with the help of microorganisms, and it makes it a salt. You remember? Salt, nitrate salts, and now the salts can be absorbed. And my pen does not want to write on that part. So, important. Nitrogen is absorbed not from the air, but from the soil for proteins, and that makes sense. But we also know that DNA and RNA are also DNA. Nucleotides are also from amino acids. Chlorophyll, it's a structure. Structure are built by proteins. Now we can see that if the plant has poor growth, and the lower leaves are yellow, and the upper leaves also starts to get pale. But the lower ones first start to get yellow and pale. Because the nitrate starts at the bottom, it has a nitrogen deficiency. And what can you do then? To add some nitrates, you can put in some manure or fertilizer. That will help. Now, the second one is magnesium. And magnesium have also your upper leaves first will look pale and yellow. But there's also chlorophyll, and it's needed for the enzyme. My plant will grow difficult. Lower leaves pale, upper leaves at first normal. Lower leaves is pale. There the lower leaves is yellow. This starts with the lower leaves, but we first see a lack of growth. In your textbook, they have a picture on page 155 for magnesium. Ne? And they say the upper leaves is normal and the lower leaves pale green or yellow. Okay. This you have in your textbook. The phosphates is a salt from phosphorus. They are important for DNA and RNA, cell surface membranes. Yes, phospholipids. You remember the phospholipids that we have in our cell membrane? You remember them? Phospholipids and the ATP. ATP, that P is phosphates, DNA, RNA, cell membranes, and ATP. And brown on the leaves, especially in your book, we see it's discoloring your leaves. Your leaves get a brownish, purplish, depending on which species of plants the deficiency is occurring in. The leaves will discolorize and it will have small leaves. Okay, now the nitrogen, the magnesium, and the phosphorus are called macronutrients. Macronutrients means that the plant needs it in large quantities. Now, that's why it's called a macronutrient. The plant needs quite a lot of these salts. Nitrates, magnesium, and phosphorus. Okay? The nitrates for N proteins, amino acids, N, DNA, RNA, chlorophyll, and auxin, that growth hormone. Now, magnesium, chlorophyll, there's chlorophyll, and there's chlorophyll. Good. Chlorophyll, and enzyme activator, and I will have the lower leaves pale. Upper leaves pale, poor growth, upper leaves pale. Lower leaves pale, well, also poor growth or stunted, short plants. Nee. Phosphorus, P, ATP, cell membrane, phospholipids. Okay, and DNA and RNA, brown colored leaves. Then iron. Now, if these are macronutrients, this one is a micronutrient. It only means that my plant needs that in small amounts, but still it is very, very important. And there we have chlorophyll again. So, nitrogen, magnesium, iron, chlorophyll. Enzymes I have again, and electron carriers. It helps with the biochemical processes that needs to take place. And again we have the upper leaves that have chlorosis. Now that word chlorosis means only that the plant lose its green color due to the chlorophyll. So chlorosis we will have in the nitrogen, in the magnesium, and in the iron. We will have chlorosis, where the leaves turn yellow due to a lack of chlorophyll. The chlorophyll cannot form when I do not have the proper minerals in the plant. Okay? I hope that you do understand that. Here I have a nice chart, and we only have to know nitrogen, phosphate, magnesium, and iron. Now, We only have to know them. This will help you. Of course, we know about carbon dioxide. Carbon dioxide is needed for photosynthesis to take place. So you can perhaps use this nice one to help you to remember. And now, how will these minerals move from the soil to the plant? The first important thing is by active transport. Mineral ions need to be absorbed by active transport. Active transport means they are normally at a very low concentration in the soil. And due to the fact that they are needed, 3 of them were macronutrients, so they are needed in large quantities. There will be a high concentration in the cells. So these ions from the soil, and they are dissolved between the soil particles in the water, they need to move via the root hair cells by diffusion. Now, this is diffusion, and specifically, sorry, not diffusion, active transport. Active transport. Diffusion means from high to low. So active transport, it needs to move upwards, and that means it needs an energy amount. It needs energy to move upwards, and that is the reason why we will have lots of mitochondria in the root hair cells. Sorry, they will be right there. Now, they will not be inside the vacuole, but inside the cytoplasm. And we draw this with a ruler. Good. To produce the energy that is stored in the molecule of ATP, so that energy can be released to move the minerals up a concentration gradient. And here we can see when it moved by active transport, it now can move through the cortex layer into the xylem. And with the xylem, it will move with the water, it will diffuse along the plant stem to the leaves. And this process of transportation in plants, we will study in our next chapter much deeper, so that we can understand perfectly. But for now, it's important to remember, up a concentration from low to high means energy is needed. Energy, ATP, active transport. Now, that is how the mineral salts will move until it reaches the xylem. Water moves by osmosis. That is also very important to differentiate. So now you can complete activity 3 on page 155 to see if you did understand this work. Then you can help your mother or father to look in your garden for signs that maybe a plant has a deficiency that can be helped with some minerals. Enjoy your gardening today, dear Clark.
What's up, world? This is Shay, the goddess of the airwaves, on 99 FM, Monday to Friday, serving you breakfast on the ignition between 6 and 9. We taught you to be inspired, taking you to master your destiny. Now it's time to do the work.
Just show up.
Put on your work boots and do the time.
Make it, and then make it better.
Hello world, it's your boy Script here on The Kingdom, where we're reminding you every single day.
Get up. You've got this. Start where you are.
Hey, this is your girl Treza on Hitspo on 99 FM, letting you know it's okay.
Try.
Make mistakes.
Ask.
I am Sibo, and I'm going to be with you for quite a while. Thank you so much for joining us here on your inspiration station, where we let you know
There's no easy money. There's no easy job. There's no easy life.
99 FM. Do the work.
Nothing is too good for my daughter. Now here's the deal, Nambi. There's no playing around with other boys before your wedding. Do you understand me? It's the most important day of your life.
Nambi, baby, you have a face like a beautiful person. I got you this Virgin Mary necklace to protect your innocence.
Stupid man. We have loved each other since high school, and now you want to turn your back on me? All the plans we made, you just throw them away? And you want Nambi now? When I've always been there for you, Ziwa, do you understand? And she doesn't even love you. I wasted my time on someone like you.
Don't be like that, Jalia.
Love, everybody needs it. Love.
beautiful souls, this is Nikita Winkler, and I am here to share easy tools for stretching that you can do just about anywhere. This is a demonstration, so what's most important is that you take the time in each position that your body needs to release its tension. In this session, we will stretch the hips. What you'll need are yoga blocks or books, thick books, or pillows, especially if you have tight hips. You can do this on a mat, on the floor, on a blanket, or even in your bed. Let's get started. So we're going to start on the floor with the soles of our feet together. I'm going to grab around the feet. This is a butterfly position. So in order to warm up the hips, I'm just going to flap the butterfly wings. And again, take your time. I'm going to lean forward. And here, again, if the hips are too tight, you can place your blocks, books, or pillows underneath. You might even feel a stretch in the back here. From this position, I'm going to take one arm right behind me, not too far from my body. The other arm, inhale, exhale, press the hip open. And come back to center. Change. One arm right behind me to lengthen the spine, the other hand on the knee. Inhale, exhale, press the hip open. And back to center. Now we're going to lie down on the floor. And first, before I get started, I have to make sure that my body, my spine is in alignment. I'm going to just open up the neck area. And you can spend as much time as you need here, allowing the force of gravity to weigh on the legs to open up the hips. You can place your blocks, books, or pillows underneath the knees. And gradually, you can take the feet even further down and spend enough time in each position. From here, we're going to go into an internal hip rotation. I'm going to spread my feet to the side, lean the knees inwards. If you have a little bit more flexibility, you don't really feel the stretch, just take your feet even further apart to deepen this internal rotation stretch in your hips. From here, I'm just going to place my feet, and without repositioning the feet, I'm going to take my arms to the side, and then I'm just going to rock my legs side to side, keeping the upper body facing up, shoulders down. And then I'm going to stop on one side, take the lower leg over the upper to add weight to deepen the stretch. Release the leg and switch. Other side, lower over the upper. And back to center. Last part, I'm going to spread my legs as wide open as I can, and I'm going to just row the boat. So what's important here is that the knees don't lift off the floor. Make sure you row both sides. Keep the knees and both bum cheeks on the floor. Alternatively, you can just lean forward. You can hang out on the pillow. Take your time on the blocks or on your books. And that is it. Thank you for joining me. Don't forget to breathe into your stretches, and happy stretching.
Corner of southern Africa lies a vast and sparse land, where endless horizons, dramatic scenery, and untouched wilderness abound. (1,500 kilometers of harsh desert coastline, a mosaic of biodiversity, sub-Saharan savanna, acacia bushland, encased by 2 deserts. Home to wildlife and wilderness abundant, yet ever at risk. This is Namibia, home to some of the most noteworthy conservation success stories across the globe. Initiatives forged, manned, and driven by passionate individuals and groups.
People are busy there walking for more than 45 kilometers a day.
Special species and wild locations under constant threat from an increasingly destructive developing world.
One of the world's true survivors.
This is the story of the Namibian wilderness, the stories of the enigmatic wildlife and natural wonders of her realm.
They are not a threat to anyone, if you understand them.
This is the story of those determined to preserve it, and thereby themselves.
Don't think I want to tell my children one day that we had rhinos in Namibia. I want to show them live rhinos.
This is the story of the last defenders.
We have been tracked them for about 7 days, and on the 7th day, we come across the butchers, and there was a shooting. We are not armed. We having a camera and GPS, we are just hiding behind the bushes. If I don't protect this species, who's going to do that? So is that why I sacrificed my life to go on conserve the rhinos.
Armed out in the field with basically a notebook against poachers that are armed with highly dangerous weapons and more. How is this an equal fight?
I started right on the ground as a tracker, and I worked all the way up to be the CEO now, and I know how hard it is. It's commitment and dedication and the patience that you have for the animal. It's really hard. It's like you had something in your house that you are protecting for so long, and when you go away from home, somebody comes and kills you.